Okay, so now we've learned a bit about what the microbiome is and about how we detect and study the microbiome. So what does a healthy human microbiome look like? Because if we want to understand how the microbiome might be affecting disease, we first need to understand what the microbiome looks like in healthy individuals. So there have been quite a few studies of healthy microbiomes looking at populations of healthy people. One of the first of these was the Human Microbiome Project. So that's at this row of the table here. This was a project that was initiated by the National Institutes of Health in the USA. Had around about 124 people. So not a huge number of people in terms of getting a great diversity there. But what they did do was look at a lot of body sites. So they looked at not just the gut, which is one that has been primarily studied in a lot of these other studies, as you can see, but they also looked at the skin, the nasal microbiome, the oral microbiome, and the urogenital microbiome. So this was a really comprehensive survey, albeit of a relatively small number of people. So around the same time, around 2008, there was also the meta-hit study, so similar numbers of people in Europe. In this case, they were looking at the gut. This has also been the American Gut Project. So this is a kind of citizen science project where people collect their own samples and then pay to have them sequenced so that they can see what is in their gut. I think they also have been collecting samples from other sites as well. So this is an ongoing project, and last time I checked, actually this number is out of date now, but this was the number from last year, there was around 10,000, and it's certainly a lot more than that now. There's also other projects, the Flemish Gut Flora Project, the Lifelines Deep Project. This is in the Netherlands. Now you might be noticing here that there's a bit of a bias in the type of people that have been studied. There's a lot of people who probably have white European ancestry within these data sets. So a lot of the information that I'm going to present to you about what we know of the healthy microbiome is based on these studies, but we sort of need to bear in mind that we probably haven't covered a lot of the diversity of people in the world, and so this may not be the case for all types of people. We are now starting to see some more large-scale studies in different populations. So there has been an ongoing study of the healthy Chinese population where we've got more than 1,000 people in that study, and also a recent study that's come out of Israel where again there was more than 1,000 people studied there. But as you can see that most of these studies, most of these large-scale studies trying to characterize the microbiome have been carried out on the gut. So really this is where we have the most information from. So the types of questions that people are asking when they're trying to understand what a healthy microbiome looks like is around stability. So how stable is our microbiome over time? Do we have a core microbiome? Is there a core set of species that really defines what a healthy microbiome should look like? Also questions around the origin and assembly of the microbiome. So where does it come from? Where do we get our microbiome from? And you may have seen in the NPR video that one of the first places we acquire microbes from is our mother when we're born. Also the effect of things like lifestyle, diet, genetics, and then of course what's the relationship between human disease and our microbiome? How does our microbiome affect our health state? So let's have a look at some of the broad characteristics that have been observed throughout all of these studies. Now, a bit of the data that I'm going to present here is from the original HMP study of only 124 people, but actually a lot of these characteristics have been repeated in larger studies. And I'll also be showing some data from the American Gut Project as well. So something that was actually already known from culturing studies, but that which was shown pretty conclusively from doing sequencing studies, is that we have really distinct communities at different body sites. So different body sites are really representing different ecosystems of microbiomes. So you can see in this plot, this is a principal coordinates analysis where each dot represents a sample. So each dot represents a whole microbial community. Dots that are closer together will be more similar to each other, and dots that are further apart will be more different to each other in terms of their community composition. So you can see that we have all the gastrointestinal samples here clustering together in blue, our urogenital samples here in purple, a little bit of overlap between urogenital and skin in green, and certainly quite a bit of overlap between the skin and the nasal samples, and that makes sense considering that it's almost contiguous between the two. And then we have our oral samples sitting over here, and again a little bit of overlap between oral and skin and nasal there. The main point being, though, that we do have quite distinct communities at different body sites. So the human microbiome is not just one ecosystem. So another thing that's been observed is that there is quite a lot of intra and inter-individual variation. So what does that mean? So intra-individual variation is variation within an individual over time. And so we do know that our microbiomes are dynamic. They do change over time, they do vary. But typically what we see is that our own microbiomes are generally more similar to themselves than to microbiomes from other people. So my own gut microbiota may change over time, but it's still going to be more similar to my previous samples than to another person. Now, the caveat to that is, of course, that the differences between different body sites are much larger than this. So the microbiota from my skin is going to be much more similar to someone else's skin microbiome than to my gut microbiome, for example. So in terms of the interpersonal variability, so this is variability between different individuals, what we see is that normally there's 2 or 3 phyla of bacteria, and so I'm talking about bacteria here because most of this data is based on 16S, which we only detect bacteria when we use that method. And phyla is a really high level of classification that's sort of just below bacteria. So this is really sort of big groupings of bacteria, and when we look at these big groupings, we see that the same types of phyla pop up over and over again. So in the gut, for example, represented here by the fecal samples, we have a lot of Firmicutes in red and Bacteroidetes in orange, and these 2 phyla typically dominate the gut. In the skin, we have a lot of Firmicutes and Proteobacteria, as well as Actinobacteria in the yellow here. And similarly in the oral, we get more of the Actinobacteria, and we also get these other groups like the Fusobacteria popping up as well. But what you will notice is that there are many individuals along the x-axis here, and that the relative proportions of these phyla can vary considerably from person to person. So we go all the way from, one person who may be, almost completely dominated by Firmicutes to people who have hardly any Firmicutes within their gut. So although we see the same types of taxa, the same groups of bacteria, the relative proportions can vary considerably from person to person. And this is also seen when we get to sort of our more, the lower levels in the taxonomy, right down to species. You're going to see quite a lot of variation from person to person. So what's driving this variation? Well, we know that genetics is one thing that can influence the variation. And what I have here is a figure from a study that was done back in 2009, and this was a study of twins, so looking at the gut microbiota of twins, their mothers, and then people who are unrelated to them. And what we have here is a measure of how similar the microbial communities are to each other. So a higher score of this unifrac distance means that they are more different. A lower score means they're much more similar. And what we see is that we see the most similarity from samples from the same individual. And then we see the next highest level of similarity between monozygotic twins. Between dizygotic twins, slightly less, although the difference there was non-significant. And then when we do the comparison between monozygotic twins and their mother, or dizygotic twins and their mother, a little less similarity again, but more similarity between children and mothers than we see between unrelated individuals here on the left. So you'll notice here that this graph actually starts on the y-axis from 0.6, so the unifrac distance, the scale goes from 0 to 1, okay? So we're not necessarily accounting for a huge amount of the variation by looking at genetics, but certainly we can see that genetics does play a role. So if you are related to someone, you're more likely to have a more similar microbiome to them than you are to someone who you're not related to. Now, of course, we also need to try and account for things like the fact that we might be living in the same environment and eating the same diet with someone that we are related to. So we also know that diet plays a pretty large role in terms of variation in our microbiome. So the picture I've got here is of Jeff Leach. He's an anthropologist who studies the Hadza, which is a hunter-gatherer community in Africa. And he's also been studying their diet and their microbiome. And what he's found is that the microbiome in these communities contains bacterial families that we don't see in the microbiomes of people from industrialized nations, right? They're rare or absent in people from industrialized nations, and they typically have very, very different diets. So industrialized nations, our diets are typically much higher in protein and fat and sugar, whereas more traditional hunter-gatherer diets have far more complex carbohydrates in their diet. So he's also done studies where he's brought researchers into the community where they eat the diet of the Hadza people, and then he looks at the change in their microbiome, and it does become much more similar to the Hadza people. And then when they go back to their normal diet, their microbiome goes back to what it looks like before. So certainly this is not the only study that have been done. There have many studies been done on the effect of diet, and we know that it has quite a large impact on the composition of our microbiome. So another thing driving variation is our environment. And so, some of the bacteria that we acquire, we're acquiring from the environment around us. So there was a study a few years ago looking at how similar people's microbiomes were where they lived in the same house as opposed to people who lived in different houses, so people who weren't related to each other. And what they found was that people who lived in houses with dogs tended to have more similar microbiomes than people who lived in the same houses without dogs, or who didn't live in the same house. And they hypothesized that dogs are acting as a bit of a vector in transferring microbes between individuals living in the house. Now, I don't know about you guys, but I do have a dog, and certainly she spends a lot of time trying to lick my face, so I can absolutely understand how this could occur. It's probably not accounting for a massive amount of variation within our microbiomes, but certainly it does have an influence. Okay, so we've talked a lot about variability and variation, but one thing that has been observed is that, so that variation is where we're looking at the composition in terms of the taxonomy, in terms of what types of bacteria are there. However, when we look at function, and when I say function, what I mean is that people have done metagenomic sequencing. So rather than just looking at one taxonomic gene, which gives you an idea of what type of bacteria it is, they sequence all of the genes and they have a look at what types of genes are encoded, and they'll try and classify them into these broad functional groups. And what we see when metagenomic sequencing is done is that function is a lot more stable than taxonomy. And so we see a lot more similarity in terms of microbial functions, which is what you can see in the bottom plot here. So different colors representing different categories of functional genes. And across the different body sites, so this is within the nose, we've got within the mouth here, in the tongue, stool for the gut. So you can see that these colors, the distribution is a lot more even than when we look at the phyla distribution in the plots above. So some of this can be accounted for by things like housekeeping genes, so genes that you find across all bacteria. However, there has also been observed that there are human microbiome specific, enrichments of types of functional genes, so things like adhesins that help bacteria attach in the gut. And also that there are genes which are enriched within specific locations. So for example, within the gut, we see an enrichment of genes for short-chain fatty acid production, synthesis of vitamins and amino acids. So we have a lot of variability, but also what we see is that we tend to have signature taxa, which most often will dominate in a particular environment. So this is not true of every single person, but on average, this is what we see. So again, we've got a bunch of different body sites here. The anterior nares is within the nose, a couple of different spots in the skin. These ones in the middle here are all different areas within the mouth. You have the stool representing the gut, and then different locations within the vagina. And you can see here, so each of these circles, the size of the circle is representing how prevalent they were across the people that were studied. And so you can see that large circles were quite prevalent. So they were found in a lot of the people studied. So for example, we see a lot of propiobacterium in the skin, propionibacterium within the skin. Things like Corynebacterium and Staphylococcus, they're really common in the skin. The individual species might vary between people, but we tend to see these taxonomic groups. Within the mouth, we see a lot of Streptococcus, we see Haemophilus, we see Prevotella and Veillonella. Within the gut, we have a lot of Bacteroides. They tend to be quite prevalent. And then within the vagina, a lot of Lactobacillus. And we're going to talk a little bit more about that in the tutorial as we have a look at these microbiome data sets from a vaginal microbiome study. So now we know some of these broad characteristics. We know that we tend to see signature taxa, there are particular phyla that are common, but within those constraints we do see variability. So how do we define what a healthy microbiome is? One thing that I think we can safely say is that we're not going to be able to define this in terms of a specific assemblage of species because there's so much variation between people. We just don't see a core microbiome, a particular group of species that's always present in healthy people. So probably we have some functional redundancy where different types of taxa can carry out the same function within a microbiome. We possibly can define it by particular signature taxa, which we think are protective. So for example, in the skin, Staphylococcus epidermidis is known to be protective against some pathogens. It actually can produce compounds which specifically inhibits things like Staph aureus. So maybe we can define it by signature taxa, but what's more reliable is measures like diversity and thinking about concepts like resilience and stability. And really these are things that are relative, right? So there's no set level of diversity that we say is the healthy level. It's always in terms of comparing to someone who, say, is sick or has a particular disease. So diversity is really referring to how many different types of microbes are there, or it can also be diversity of types of gene functions encoded. This concept of resilience and stability is really about whether the community can bounce back from a disturbance. So we've got a couple of graphs down here where if you have a stable community, you might envisage it as being sort of down in a ditch like this where you might have a perturbation. It might push the community up to here, but it's going to be able to pretty safely return back to where it was after that disturbance is removed. So in the context of the human microbiome, that might be something like taking antibiotics, which is obviously going to wipe out a bunch of bugs in your gut. Now, if you have a disturbance that's really large and pushes you into an alternative state, then it might actually be quite hard to return to your original state. So a community that's very resilient is one that's able to get back to this original state, and one that is not so resilient is one where it's really easily pushed out of that equilibrium. It's pushed into an alternative state. And so things like diversity are one of the markers that we look at because typically a more diverse community is more resilient and stable. Now, that's not necessarily true in every body site. It's definitely something we see in the gut. But as you'll see when we look at the vaginal microbiome, almost the inverse is true. When we have a less diverse community, that tends to be more stable. And we'll talk about why that is when we have a look at the data. Okay, so just summing up what we've talked about here, different body sites have distinct microbiota. We see a lot of variation due to things like diet and genetics and environment, but we do see particular signature taxa that we know are quite prevalent within different body sites. And when we're thinking about what a healthy microbiome is, it's less thinking about particular types of species and more thinking about whether our microbiome is resilient and diverse.